Good morning, everyone. My name is Laura Nilsson. I'm the Associate Dean for Graduate Education in the Faculty of Science. And on behalf of myself and the Dean and our entire faculty, welcome to the 32nd edition of Soup and Science. Before we begin, I would like to acknowledge that McGill University is on land which long served as a site of meeting and exchange amongst Indigenous peoples, including the Anishinaabe and Haudenosaunee nations. We acknowledge and thank the diverse Indigenous people whose footsteps have marked this territory on which peoples of the world now gather. Today we have 5 incredible speakers. We have Professor Simone Caron-Huot from the Department of Physics, Professor Galen Halverson from the Department of Earth and Planetary Sciences, Professor Masha Prager-Khoutorsky from the Department of Physiology, Mr. Benjamin Rutzky from Computer Science and Biology, and he's a very special speaker because he just started his PhD in the Quantitative Life Sciences program, but he did his undergraduate research at McGill, and that's what he's going to be talking about today, excuse me, to give you an idea of what undergrad research really looks like. Finally, we'll close with a speaker, Professor Amy Blum from the Department of Chemistry. Following the presentations, the students in the audience will have the opportunity to answer a skill testing question about each presentation. And the top 10 students who answer the questions correctly in the fastest time will win a prize, which will be a $25 credit on your student card toward the purchase of a meal at any on-campus dining location. So this is really helping us, even in the remote context, to put some of the soup potentially back into Soup and Science. After this competition, we're going to move to the breakout rooms where we'll have a moderated question and answer session with our speakers. We'll have one speaker and a moderator in each breakout room. You'll be able to choose which room you want to visit and join the breakout room on your own. The way this is going to work is that a few minutes, we'll have those discussions, and then a few minutes before noon, we'll return back to the main session for final remarks. With that, we will now begin with our first presenter, Professor Simone Caron-Huot from the Department of Physics.
Everyone, I'm trying to share my screen. I think you have to stop yours. Thanks. Great, yeah. So welcome everyone. So I'll tell you briefly about one idea that's important in my work. Then I tell you about what I actually do. Is the sound correct? Yeah. So causality in modern physics is not just that you can't influence the past, but also it takes time to influence a faraway place. So we say that information cannot travel faster than light. And this is an idea that explains a lot of things. So for example, if you have electric charges in an antenna, these charges exert a force on the charge in a receptor. This force is small, who cares? But if you accelerate the charge in the antenna, if you vibrate it, then the delayed information that comes through a detector has to come along with energy. And these are the electromagnetic waves. And so they inform the world that the particle has accelerated. So the world, in fact, is quantum mechanical. So energy comes in discrete packets that are called particles. So this very general principle of causality explains why all forces in the world are mediated by particles. The antenna emits photons. In particle physics, we explore how things work at small distances. So we know that we're made of atoms, which are very small. But the atoms are made of electrons running around nuclei. The theory of the electron didn't make any sense unless it has an antiparticle called the positron, which was found. The forces between the nuclei didn't make sense if they were not mediated by new particles called gluons, which were found. But the interactions between those didn't make any sense if they were not mediated by what we call the weak bosons, which were also found, along with a whole bunch of unexpected stuff. Okay. we don't predict. There's a lot of weird things happening, but then it turns out that the interactions between those things didn't make any sense if there was not an extra force carrier called the Higgs boson, which was discovered in 2012. But now the weirdest thing happened. For the first time in history, it all makes sense. Well, it doesn't quite all make sense. We're still missing gravity, and we're still missing a whole bunch of stuff that we don't understand, like dark matter, neutrinos. There are things that neutrino masses that don't really fit into this. But for the first time in history, there's no immediate problem that requires an immediate resolution. So what I do? So I'm a theorist. So my job is to convert coffee into formulas. And so one of the line of things that I do is to try to do precision calculation, where we look for cracks on the standard model at experiments that we can do that probe this length scale. And in fact, I'm actually over time. So yeah, I'm just saying here that we can use strong computers, but can also use the most basic principles, and they also help us a lot in doing these calculations. And the other thing I try to do is to understand how gravity fits in this picture. So thank you all, and see you in the discussion.
Thank you, Simone, for that great presentation. Next, we have Professor Galen Halverson from the Department of Earth and Planetary Sciences.
All right, hello everybody. I'm happy to be here. And so what I'm going to talk to you today about is how geologists can actually interact with biology and biologists to inform what we know about the early evolution of life on Earth, and specifically the origin and diversification of the eukaryotes. Okay, so this is a bit of a busy figure right here, but what you see on the right is a phylogenetic tree of eukaryotic life. My main point here is to show that a lot of the key diversifications amongst eukaryotes happened in the Precambrian. That is well before the first sort of macroscopic fossil record of animals appeared, about 570 million years ago, with the appearance of these strange and wonderful Ediacaran biota, to be followed by the Cambrian radiation shortly thereafter. What I'm going to do now is sort of step back a bit earlier in geological time and talk about how geology tells us a bit about how life evolved. So for one, we can contemplate the origin of life on Earth in the first place. And most scientists now agree that probably it could not have originated and taken a foothold prior to about 3.9 to 3.8 billion years ago, when Earth was being heavily bombarded by meteorites and continually sterilized. And we know about this from studying the record of meteorite impacts on the moon. And now we think geologists generally agree that the best early evidence, unambiguous evidence for life on Earth comes in the form of stromatolites. These are these really cool lithified, sort of organically layered structures that are formed through the interaction of photosynthetic bacterial mats and sediments and seawater or lake water. And the oldest of these are about 3.5 billion years old. So we have bacteria by 3.5 billion years ago, and we probably have archaea by around the same time, if not earlier. And we get the evidence for the earliest archaea is more cryptic, and it comes in the form of isotopic fingerprints, these very unusually negative carbon isotope fingerprints in methane and other biological materials that date that far back in time. Now, the next sort of clues don't come for quite a bit later, until about 2.4 billion years ago, with the so-called Great Oxidation event. This records when oxygen first existed in the atmosphere freely. And we see geological evidence for this in the form of red soil. So for the first time, iron oxides were forming on the continents. And so this implies that by this time, oxygenic photosynthesis by cyanobacteria or their close relatives had evolved. Probably it evolved earlier, but for sure by about 2.4 billion years ago. So the next big questions are how the eukaryotes evolved in the first place, and then how the algae evolved. And both of these events were symbiotic events. So eukaryogenesis, the origin of the eukaryotic cell, it's generally agreed upon now was the consequence of a symbiotic merger between an Asgard archaeal host cell and a bacteria, an alpha proteobacteria that became the mitochondria. That is the metabolic engine in the eukaryotic cell. The questions arise when we try to understand this process of eukaryogenesis from sort of the first precursor known as the FECA, the first eukaryotic common ancestor of the eukaryotic cell. It's the last eukaryotic common ancestor that had really all the bits and pieces that we recognize as being eukaryotic. And then this cell then would have given rise to the great diversity of eukaryotes today, including mushrooms and trees and corn and us and lots of different protists. So the question is, how do we actually identify early eukaryotes in the fossil record? And the short answer is not very easily. But we have a few criteria that we use. For example, large size, that's relatively large size because these are microfossils, complex cytoskeletons and ornamentation, and evidence for sexual reproduction in the form of what we call existent structures. That is basically the cleavage of cells to allow spores out. And these are just some of the fossils that we think represent some of these early Phyca to Leca transition organisms. And you can see they have some interesting structure on their cell walls, and there are examples of excitement as well. We don't know a lot about what these are or the habitats in which they live, but we know they date back to about 1.7 billion years ago. The first taxonomically identifiable eukaryotic fossil comes in much later. They're found in these rocks, in these cliffs on the north coast of Baffin Island. In the form of these really quite spectacular detailed microfossils that have been identified as the red alga Bangiomorpha. And very recently, we are actually finally able to date these through so-called radiometric techniques to 1.05 billion years old. And this is a very important date because we can combine this with molecular phylogenetic data to generate a molecular clock and actually date the origin of the algae. That is the endosymbiotic event that gave rise, that is an endosymbiosis between a eukaryotic cell and a cyanobacteria that gave rise to photosynthesizing eukaryotes. And this happened about 1.25 billion years old, 1.25 billion years ago. And we'll just end on the note of discovery, and that is that this same molecular clock suggested that the green algae would have originated after about 1.1 billion years ago. And we just discovered what we think are the oldest, or amongst the oldest macroscopic green algal fossils that are about 0.95 billion years old, and we just published this study recently. So there's still a lot of room for discovery in the study of early life on Earth. Thank you.
That's very interesting. Thank you. It's amazing to see those images of such unbelievably old creatures. Next, we'll go to Professor Masha Prager-Khoutorsky for our next presentation.
Hello, this is my lab. We are located in the McIntyre Medical Building. We are interested in how brain regulates basic function of the body, such as hunger, thirst, and cardiovascular function. The brain area responsible for this regulation of this function called hypothalamus. It's a small region located in the base of the brain. Hypothalamus in humans has the size of the nail thumb. It can detect metabolic changes such as increases in glucose levels after a meal and regulate hunger and satiety. It also regulates hydration status of the body by triggering thirst. It controls function of different organs such as heart, stomach, kidney, and blood vessels via regulation of autonomic nervous system, and also controlling sleep, body temperature, and sexual and reproductive behavior. This is done by constant bidirectional communication between neurons in the hypothalamus and peripheral tissues and organs. However, most of the neurons in the brain or the hypothalamus cannot directly communicate with the periphery due to the existence of the blood-brain barrier, a protective barrier structure that facilitates the entry of nutrients and oxygen from the blood vessels into the brain, while preventing from other substances such as pathogens and certain drugs from entering into the brain tissue, creating a very stable, nearly pathogen-free environment. However, there are a few tiny brain areas called circumventricular organs lacking this barrier. Thus, neurons in this area can directly send signals in the blood. What makes circumventricular organs so special and different from the rest of the brain is that they have leaky blood vessels, and thus blood-borne molecules can enter the brain. And they also have specialized glial cells called tinocytes. This is my favorite circumventricular organ called OVLT. It has shape of the heart, and it regulates cardiovascular system and sexual and reproductive behavior. Tinocytes are these pink cells that create the heart shape. These cells are very special because they have a cell body or a head located in the brain and exposed to brain environment and long processes or legs, if you wish, with feet interacting with leaky blood vessels and thus exposed to the peripheral circulation. Tinocytes can sense and respond to signals in the blood, such as glucose levels, and modify the leakiness of blood vessels and thereby regulate if signals from the body will or will not arrive to the brain, and vice versa, if signals from the brain will reach the periphery. Studies suggested that tinocytes play a role in the regulation of energy metabolism and might be implicated in obesity and diabetes. The function of the cells in the OVLT is not known, and we study if OVLT tinocytes are involved in the regulation of cardiovascular system, fluid balance, thirst, and their potential role in cardiovascular disorders such as hypertension. Thank you very much for your attention. I'm looking forward to the discussion.
Fantastic. Thank you. Very interesting and really striking images. Next, our next speaker will be Mr. Benjamin Rutzky, who will tell us about his undergrad research as a student in computer science and biology.
Okay. So, ants are social insects who live in colonies consisting of many female workers and a queen who spends her life laying eggs. In order to give rise to new colonies, some of these eggs develop into winged reproductive males and female future queens who fly away to mate with ants from other colonies. Scientists have many questions about the timing of these ant nuptial flights. Prior research has looked at possible connections between environmental cues, such as temperature. Oh, sorry, can people see my slides?
They're not moving forward. We can see the slide, but they're not transitioning.
Yeah, no, I only have this one slide.
Do they have that one? Okay.
Yeah. Okay. So, yes, so prior work has looked at the connection between the timing of these ant nuptial flights and certain environmental cues such as temperature, precipitation, and pressure. And while these works were groundbreaking, they were also limited because they only focused on a few species over a very brief period of time. In order to draw conclusions for the more than 15,000 species and subspecies of ants, we need a lot more data. To this end, over the past 3 summers, I have worked under the supervision of Professor Ehab Abuhef from the Department of Biology to develop and redevelop AntNupTracker. This app allows citizen scientists and professional myrmecologists to browse and contribute to a completely open source database of ant nuptial flight records. This app lets users report nuptial flights when they see them from the field, and there is really no minimum level of expertise required. The flight recording process is designed to be very friendly to novice and flight recorders. For example, for help with species identification, users can upload images of flights. Other users can then leave comments to suggest what the correct species is, and a professional myrmecologist can come along and mark the flight as verified once the identification is correct. It's also important to note that as soon as the flight is recorded in the app, our server fetches the weather information for the location of the flight to facilitate future analyses about the links with weather. All of the data reported in our app is completely open source, so anyone can use it for their research as long as they give credit to the users who originally recorded the flight. In addition, our entire app and server backend are open source, enabling as many people as possible to get involved studying ants either through recording nuptial flights or through helping to develop and improve our app. In July of 2020, we released an iOS version of the app. And this past summer, in research funded by an NSERC USRA award, I completely redeveloped the app using the Flutter framework developed by Google. This enables the app to be easily translated into many languages and to run easily on both iOS and Android. So these improvements will enable us to hopefully reach as many people around the world as possible and get as many flight records as possible. In a few years, we'll hopefully be able to start looking for patterns between nuptial flight timing and weather, and we'll be able to also observe the effects of climate change. Down the road, we may also be able to mathematically model the timing of nuptial flights and be able to use AI to predict when and where flights will happen. And this will also enable ant collectors to find newly mated queens to collect instead of having to uproot existing colonies, which is destructive to ant habitats. So while this project is still in its relative infancy, the possibilities are truly endless. Thank you.
And thank you, fascinating, really, really interesting. It's great to see more citizen science. So our final speaker is Professor Amy Blum from the Department of Chemistry. Go ahead, Amy. Amy, are you? I don't hear you.
Better?
Yeah, there you go.
Okay, good. All right. So I'm going to tell you a bit about our some of our work in metamaterials, which are materials that have properties that are not found in nature. So one of the goals of this project is to figure out new ways that we can make optical materials. And one of the reach goals for this project is to come up with new ways that we can make things invisible, which has been a long-standing dream of humans, from Harry Potter to the cloaked Klingon armada to militaries everywhere. People would think it would be really cool to make things invisible. The problem is that with currently existing materials, we can't bend light in a way that would actually make true invisibility work. So what actually happens when something is invisible to our eyes? When we see, what we're actually seeing is light that's bouncing off of an object. So we see the light after it interacts with the object. In order for an object to be made invisible, what we would want is for light to pass around that object in such a way that the light didn't get scattered off the object, so we can't find the light that's interacted with it. So people have come up with ways to do this involving other forms of light other than visible light. This is an experiment that was done in 2013 that works in the microwave region. And so the idea is they made these structures out of gold. They're millimeter size, it's about 7 millimeters or so in size. And they make these individual ring structures. They print them on flat 2D sheets, and then they take these 2D sheets, stack them up into 3-dimensional structures. And what does it actually do? On the bottom here, what you see is a field of straight lines. These are microwaves. When you stick an object into this microwave field, just like with visible light, the light waves bounce off of it, they interfere with each other, they get scattered, and a microwave detector can see the object. When we make these kind of resonators that cloak, what happens is the microwave field is distorted a little bit, but the object that's in here, you can't tell what size it is or what shape it is. So it's like, when you see in Star Trek with the Klingons, how it's a little bit blurry, but you can't see that they're there. That's the same idea, only for microwaves. So this works actually pretty well, but it only works in the microwave region. So you can hide things from a microwave oven, but not from a human being. So what my group is trying to do is take this kind of work and bring it to the visible wavelengths, which requires a reduction in size from millimeters to nanometers. So the way we go about using this is we use viruses, which are inherently nanometer sized, as a template for making our structures. So the advantage of using a virus is that we have site specificity. We know what all the chemical groups are on the surface of the virus, so we can then use chemistry to stick things on there. So our plan is to take 4 billion years of natural selection to make a stable, chemically resistant shell and make it work for us. And so the idea is we're going to take this virus, tobacco mosaic virus, that makes these disc-like structures that look like little donuts. And we're going to use these little donuts to make rings and then build these rings into sheets and 3D structures. So does it work? Well, I wouldn't be showing you this if it didn't work at least a little bit. We can make these 1D point structures from rings. And what you're looking at here are transmission electron micrographs showing gold nanoparticles around a central disk of virus, which you can't actually see because it's a light material. Recently, we've been able to take these ring structures and transform them into 2D sheets. And under the right conditions, we can take these 2D sheets and curl them up into 3D rods. And so we take our ring structure, make 2D sheets and then 3D structures. And now what we're doing is we're measuring the properties of them to see if they would be useful to make things invisible, among other sorts of things that we might want to do. And in terms of undergraduate research, I would like to point out that an undergraduate student took our recipe for gold particles where we used to only make 3.5 nanometer gold particles. And it was an undergrad researcher who figured out how to make them at 10 nanometers and 16 nanometers in a controllable way. So thank you for your attention. And I'd be happy to discuss it more in the discussion.